I am going to present the in vitro propagation of an endemic medicinal orchid, Anectocailus elatus. Objective outline of my study is about introduction, objectives, materials and methods, results and conclusion. And coming to the introduction to plant tissue culture. Plant tissue culture is nothing but a collection of techniques that is used to maintain, grow plant cells, tissues or organs under sterile conditions on a nutrient culture medium of a known composition is known as a plant tissue culture. And it is a widely used technique that is used to produce clones of a plant in a method known as a micropropagation. The person in this picture is Gottlieb Haberland. He was an Austrian botanist and he was the first person to introduce the plant tissue culture. And some of the applications of this plant tissue culture is to conserve rare or endangered plant species and production of identical sterile hybrid species and removal of viruses by micropropagation from meristematic tissues. And there are three main steps involved in the plant tissue culture. First is the stage 1, that is the initiation phase. Initiation phase, it's nothing but any type of explants when cultured on a medium is known as, that is leaf, internode, stem, etc. were cultured on a medium is known as an initiation phase. And stage 2 is the multiplication phase. Cytokines play an important role in the multiplication phase. And cytokines such as BAP, TDZ and kinetin are used for this multiplication phase. And stage 3 is the root formation phase. Auxins play an important role in the root formation phase. And some of the auxins such as NEA, IAA and IBA play an important role in this root formation phase. And media are of different types. One is an MS medium, that is Murashik and Skoog medium. It was found out in 1962. And second one is Mitra orchid medium. It was found out in 1976. And Linman orchid medium, it was found out in 1970. These are the different types of media that is used for inoculation purpose. And germination is of two types. One is in vitro germination and another one is ex vitro germination. And in vitro germination, it includes two things. One is a symbiotic seed germination and another one is an asymbiotic seed germination. Symbiotic seed germination, it's nothing but germination of seeds that takes place with the help of fungal association. And asymbiotic seed germination, the germination takes place without any help of fungal association. Next slide. And these are some of the plant tissue culture techniques in orchids. And the first thing is the green pot culture. It's nothing but when immature seeds from the green capsules, that is prior to the dehiscence, are cultured in vitro on a medium. Second thing is an asymbiotic seed germination. It's a basic and an important tool for propagating a large number of orchids and hybrids. And third thing is a nodal culture. It's a very simplest and the basic method of micropropagation, which is used to regenerate a large number of plants within a short period of time. And this is some of the general introduction about the orchids. And orchids is the largest family of the flowering plants, and it consists of about 850 genera and 35,000 species, and is distributed worldwide, that is India, Sri Lanka, South Asia, South and Central America and Mexico. And orchids are of two types. One is an epiphytic and another one is a terrestrial orchid. And orchids are also used as medicinal, food, and also as ornamental and horticultural. And some of the orchids that are used ornamental and horticulture are Vanda, Dendrobium and Cymbidium. And some of the orchids that are used as food are Anectocailus. The leaves of this species is used as a leafy vegetable. And Diuris, it is a sweet tasting tuber. And Orchis, this tuber, it is used for preparing milk custard. And some of the orchids, it is used medicinally also. Pholidata, the pseudobulb is used in curing abdominal pains and also in rheumatism. Vanda, the flower juice of this is given orally for dysentery as well as diarrhea. And the flower juice also is used to cure many skin diseases. Malaxis, it's an important medicinal orchid, and this it is used in the preparation of Ayurvedic tonic, that is Chyawanprash. Next slide please. And this is a schematic representation of some of the medicinal uses of orchid. And this picture is Dendrobium flower, and that is Sarcanthus flower. And these are some of the uses that is used in ornamental purpose, food, and also as medicinal. Next. And this is my present work. And Anectocailus elatus, it's a rapid and efficient method for mass propagation from the nodal explants was developed. And this is a habit of this species. The leaves of this is usually velvety maroon green or blackish green with a glistening metallic copper vein, silver or gold. This is the flower of this species and it blooms during November to December. And this is a general introduction about the Anectochilus. And the genus Anectochilus, it comprises of about 50 species and it is a very small terrestrial herb and it's a shade growing plant and it usually grows on leaf litter and also in humus and on rocky rocks. It is distributed in Himalayan foothills in India to mountains of Southeast Asia, Japan, Indonesia and some of the Pacific islands including Taiwan. And this orchid, it is commonly known as the South Indian jewel orchid because of its attractive foliar venation. And this orchid is also used medicinally, that is to fever, pain, lung and liver diseases and also in underdeveloped children. And some of the active compounds has also been isolated from this genus, that is quincinolide, butanolide and guduracide. Coming to the objectives, to standardize an efficient protocol for mass propagation of Anectochilus elatus from the nodal explants and to study the effect of various plant growth regulators on mass multiplication and establishment of in vitro raised plantlets. Healthy explants were collected from the Valparai Hills in the southern western Ghats of Tamil Nadu, India, which is located at 10.37 degree north and 6.97 degree east. The collected species was identified and the voucher specimen was deposited at BSI, that is Botanical Survey of India, Southern Circle, Coimbatore, India. The existing number of this species is this thing. And the nodal explant was selected for micropropagation. Next slide. Effect of auxins. The explants were cultured on MS medium, that is half strength, supplemented with various auxins such as NAA, IAA, IBA, 2,4-D and TIBA, that is to 0.25 mg to 2 mg per L, either individually or in combination. And the above medium was supplemented with 3% of sucrose and it was solidified with 0.8% of agar. And the pH, it was adjusted to 5.6 to 5.8. This slide is about the effect of auxins. The explants were cultured on MS medium half strength supplemented with various cytokinins such as BAP, kinetin and TTZ, that is 0.25 to 2 mg per L, either individually or in combination. The above medium was supplemented with 3% of sucrose and solidified with 8% of agar and the pH was adjusted to 5.6 to 5.8. This slide shows the effect of combinations of multiple shoot induction. For multiple shoot induction, the nodal explants were cultured on MS medium half strength, that is supplemented with BAP 0.5 mg per L plus NAA 0.25 mg per L along with the addition of IBA, that is 0.1 mg per L either individually or in combinations. And activated charcoal and GA3, it is used as an rooting medium and this GA3 it is used for shoot elongation as well as for shoot multiplication. Next slide please. For hardening, vermiculite is used, that is the healthy plantlets were gently removed and it is washed off with the help of sterilized distilled water to remove the adhering media, and the transferred plantlets were transferred into potted plastic plants containing vermiculite, and this transferred plants were was irrigated with MS medium basal salt solution containing 3% of sucrose, and it was done every 3 days for 4 weeks, and then the plantlets was acclimatized and the survival rate was about 98%. This is a statistical method of my study, that is the differences between means and the treatments were scored with the help of Duncan's multiple range test. This is coming to the result. This is the flower of this orchid species, and the nodal explants were cultured on MS medium devoid of plant growth regulators. There is no response in this medium was observed. Next slide please. And this table, it shows the effect of auxins. Of the various plant growth regulators tested, NAA at 0.25 mg per L was found to be best for inducing maximum number of multiple shoots, that is 4.00 plus or minus 1.00 with a shoot length of 1.86 plus or minus 0.45, followed by TIBA at 1 mg per L. It shows a maximum of shoot length, that is 3.00 plus or minus 1.00 with a shoot length of 1.80 plus or minus 0.40. Next. This is a figure with the addition of activated charcoal and GA3. Activated charcoal was added to remove the phenolic exudates, and GA3 it is used for shoot elongation as well as the shoot multiplication. And this figure it shows formation of multiple shoots that is done with the help of BAP 0.5 mg per L plus addition of NAA along with IBA at 1 mg per L was induced for shoot multiplication phase. Next. This table it shows the effect of cytokinins. Of the various plant growth regulators tested, TDZ was found to be best for inducing maximum of shoots, that is 6.00 plus or minus 1.00 with a shoot length of 5.56 plus or minus 1.02, followed by BAP at 1 mg per L showed a maximum of shoots, that is 4.00 plus or minus 1.00 with a shoot length of 2.00 plus or minus 0.36, whereas increase in addition of kinetin, it leads to lower proliferation rate and also a diminutive growth. Next slide please. This table it shows the effect of activated charcoal and GA3 on different growth regulators on multiple shoot formation. Of the various plant growth regulators tested, half strength MS medium at 1 BAP plus 0.25 NAA with the addition of 0.5 gram of activated charcoal and 1 ml of GA3 induced maximum of shoots, that is 13.66 plus or minus 1.52 with the shoot length of 8.63 plus or minus 1.46. Next slide please. This table it shows the effect of combinations on multiple shoot formation and root formation. Of the various plant growth regulators tested, half strength MS medium at 0.5 BAP plus 0.25 NAE with the addition of 5 gram of activated charcoal, 1 ml GA3 along with the addition of IBA was found to be best for inducing maximum number of multiple shoots, that is 6.66 plus or minus 1.52 with the average shoot length of 8.80 plus or minus 0.40 and with the root length of 2.50 plus or minus 0.75. Next slide. And this figure it shows the small hair-like structures and addition of formation of root and shoot. Here a small hair-like rhizoids are seen and activated charcoal is added to remove the phenolic exudates, and GA3 it is used for shoot elongation as well as for shoot multiplication. And this is a hardening stage. It is done with the help of vermiculite, and it was maintained in the plastic cups under the culture condition, and after that the plantains were acclimatized and further process was carried out. Coming to the conclusion, during the field study, it is observed the species of Anatocalis elatus is facing many threats due to human interference in forest areas, cattle grazing and careless collection by the local people. Hence, the present study was attempted to conserve this endemic medicinal orchid by mass multiplication. It is concluded that the in vitro regeneration protocol established from the present study can be employed for propagation of this orchid in large scale, which may not only help in addressing the urgent needs for effective conservation, but also meet the high pharmaceutical as well as ornamental requirements. Thank you.
The dim, which was isolated from the cruciferous vegetable in the form of glucosylated form, and then and then it was quantified and then idealized to get this particular active drug, which showing the concentration dependent promostigotes reduction, as well as the this is the Leishmania trophica in the 3 different varieties of Leishmaniasis. Next one is, I'm sorry for the poor visibility of this promostigotes. You can see, actually you'll find that the apoptosis happening, actually you can see there, sorry for this. So here DIM induces apoptotic events in the Leishmania cells. That is how it is killing the parasites. So apoptotic cells detected by flow cytometric analysis showed using the annexin V and the PI in the FL and the FL2 channels shows that the B, this is the B, and then followed by bar diagram, the same one. Apoptotic cells and necrotic cells, you will find that both are safe. So topoisomerase is the enzyme which are responsible for the cell growth by in the cell divisions. What does it do? It cuts the single strand DNA as well as the double strand DNA and realligate and rejoin actually, which is how the coiling, winding, unwinding of the DNA is taking place. So fine, there are two types, topoisomerase 1 and topoisomerase 2. So one is ATP dependent, another one is not ATP dependent, etc. So what we have compared with this is to find out the mode of action, the target of the action is what we have seen that this is a simple condensed molecule from cruciferous vegetable, which is naturally it is being produced in the stomach after consuming the vegetable. So, and the camptothecin, which is a well-known anti-cancer drug. So, well-known topoisomerase 1 poison, and then this is the one which we have compared with this. You can see that this is the this is the one which is actually the unwinded form. This is the this is the cutting. Cutting is happening here. The enzyme is cutting the DNA into small pieces. This is the normal one. So this is the normal process actually. It has to happen for the cell division. So camptothecin, you will find that in a dose dependent manner, which is restored actually. So by inhibiting the enzyme activity of the topoisomerase enzyme activity, and then DIM. Also we found that the same way actually, starting from 1 micromolar to 20 micromolar. Next one is so DIM and novel DNA topoisomerase 1 poison with a strong binding affinity to the enzyme. We have found out, we have also determined the binding affinity of this molecule with this. You can see that actually completely restored. Next one is so this is also you can see that the enzyme DIM, not able to see it, I'm sorry for this poor so this is in fact stronger than camptothecin actually. It inhibits better than the camptothecin. You can just see that actually 10 micromolar of the dose with the topoisomerase Leishmania donovani topoisomerase 1 B, which is being completely restored to the normal one at this particular concentrations. Next one, it's fine. So this is having a stronger binding affinity in the dose depending manner. Actually this is the previous one. So this is the previous one. This is the non-competitive. We have to find, this is the typical graph you will find actually, if it is a competitive inhibitor or non-competitive inhibitor or uncompetitive inhibitor, you will find a typical graph. It shows that, proves that it's a non-competitive inhibitor. Next one is. So during the course, we also developed some of the resistant strain by using continuously exposing the DIM, and then it is undergoing the random mutations at different levels. This is the major. The Leishmania donovani topoisomerase has got a 1 has got a 2 binding site. One is the major groove, another one is the minor groove. So it undergoes. It is actually targeting the minor one. So next one is these are all the resistance strains. Where are the mutations happening especially, and then we have also chemically. Next one. Yeah, that's from next one. Yeah, we have also chemically synthesized a number of derivatives of modifying this DIM is naturally they obtained in a phytochemical with a lot of other by using a computational input, and we found that this is the diphenyl 2,2' diphenyl diinvalide methane DPDIM was found to be excellent even resistance strains also. That's it. I think I'll skip these things. Go to the next one. Yeah, fine. For previous one. Yeah. So in summary, what I would like to say that uptaking cruciferous vegetable means you are getting high volume of vitamin, folic acid, minerals, and fiber apart from your medicine. As a group, the cruciferous vegetables are simply superstars. Health benefits of cruciferous vegetables is associated with the prevention of metabolic disorders, etc. Dietary uptake of these vegetables causes lower risk of, lung and breast cancer. The result of our study indicate that the DIM, a phytochemical from cruciferous vegetable, poison Leishmania donovani topoisomerase 1. This is how it acts by killing the parasite, which is a class 1 non-competitive inhibitor. DIM accumulates, which I skipped actually, remains in the same, wild type as well as the, resistant parasites, no problem at all. We have also chemically modified the natural one with the different substituents and found that DPDIM found to be a better one for even resistance strains. And of course, the docking study also actually supported our findings. And next one. Yeah, next one. Yeah, previous one. Some of this, this is the publications from some of the publications from where I am. This is our publications from where I talked the presentation today. The next one is this work will not complete unless otherwise I acknowledge really the people behind the studies. Actually, Dr. Hemanta Kumar Majumdar of the infectious disease, and this is all my scholars who are actually working on this and various other disease models. And finally, the funding from CSR as well as DBT. As I promised that initially, that how to, if you fry the cruciferous vegetables and eat it, actually nothing, you will get nothing actually. Absolutely. What we are also having a problem that, in course of disinfecting the vegetable we normally in South, I think everywhere it's happening that, we just boil the vegetable and remove everything and then we eat, thinking that, we are actually making it a complete one, and then fry it. Actually, that should not be there. Only the steamed one retains all this nutritious value as well as the medicinal compound which I have talked about, which we have also shown that it is the steamed one which is actually the vegetables are good for the health. Thank you very much.